Hello, everyone. Welcome to our module on adaptive immunity. In the previous module, we examined innate immunity, the body's rapid first line of defense against infection. While innate immunity is highly effective, it has important limitations because it responds to broad patterns shared by many pathogens. In this module, we'll explore adaptive immunity, a system capable of recognizing specific pathogens, generating targeted responses, and remembering previous infections. These abilities form the foundation of long-term immunity and explain how vaccines protect us from disease. By the end of this module, you should be able to differentiate between innate and adaptive immunity and explain how antigens and epitopes allow the adaptive immune system to recognize specific threats. Before we can understand adaptive immunity, we need to briefly review innate immunity. Innate immunity is the body's first line of defense against infection. It is present at birth and responds rapidly, often within minutes or hours of encountering a pathogen. Unlike adaptive immunity, innate immunity does not recognize specific microbes. Instead, it recognizes common features that many pathogens share. For example, immune cells can detect molecules commonly found on bacterial cell walls or viral genetic material. Because innate immunity recognizes broad patterns rather than specific pathogens, the response is generally the same each time a microbe is encountered. The innate immune system does not improve with repeated exposure and does not generate long-term immunological memory. Examples of innate immune defenses include physical barriers such as the skin, chemical defenses such as stomach acid and antimicrobial enzymes, and cellular defenses such as macrophages and neutrophils that engulf and destroy microbes. Although innate immunity is extremely important, it has limitations. Many pathogens have evolved ways to avoid or overcome these defenses. This is one reason why a second, more specialized system, the adaptive immune system, is necessary. Now, the adaptive immune system differs from innate immunity in several important ways. Rather than recognizing broad patterns shared by many microbes, adaptive immunity can recognize specific molecules associated with a particular pathogen. This specificity allows the immune system to respond to pathogens it has never encountered before. Instead of mounting the same response every time, adaptive immunity tailors its response to the unique characteristics of the invading organism. There is a trade-off, however, because adaptive immunity must identify the threat and activate the appropriate cells. The first response is slower than innate immunity. It may take several days to reach its full strength. The major advantage of adaptive immunity is memory. After an infection or vaccination, some immune cells remain in the body for years or even decades. If the same pathogen is encountered again, these memory cells respond much more quickly and effectively. This ability to learn from previous encounters is one of the defining characteristics of adaptive immunity. To understand adaptive immunity, we first need to define an antigen. An antigen is any molecule that can be recognized by the adaptive immune system and trigger an immune response. Antigens are often found on the surface of pathogens such as bacteria, viruses, fungi, and parasites. They are commonly proteins, although other types of molecules can also function as antigens. One way to think about an antigen is an identifying feature of a pathogen. Just as people can be identified by unique characteristics, microbes carry molecules that help the immune system distinguish them from the body's own cells. Importantly, different pathogens have different antigens. This allows the adaptive immune system to recognize and respond specifically to one pathogen while ignoring others. The ability to recognize unique antigens is what gives adaptive immunity its remarkable specificity. Although we often talk about the immune system recognizing antigens, immune cells usually recognize only a small portion of an antigen. The specific region is called an epitope. Think of an antigen as an entire face and an epitope as a specific facial feature, such as a nose or an eye. The immune system is not necessarily recognizing the whole structure at once. Instead, it often recognizes a particular molecular shape, within that structure. A single antigen can contain many different epitopes. As a result, different B cells and T cells may recognize different parts of the same pathogen. This level of specificity is one of the defining characteristics of adaptive immunity. Each lymphocyte is capable of recognizing a particular epitope, allowing the immune system to target pathogens with remarkable precision. Next, we will ask, how does the immune system know what to attack? A major challenge for the immune system is determining what should and should not be attacked. Immune defenses must recognize harmful invaders while avoiding damage to the body's own tissues. The innate immune system accomplishes this using pattern recognition receptors, or PRRs. These receptors detect molecular patterns that are common among many groups of microbes. For example, certain PRRs recognize bacterial cell wall components or viral genetic material. Adaptive immunity takes specificity much further. Instead of recognizing broad patterns, B cells and T cell receptors recognize specific epitopes on antigens. This allows the immune system to distinguish among different pathogens with remarkable precision. The ability to recognize specific foreign molecules while tolerating self tissues is essential for effective immune function without immune system disorder. Now, we began by comparing innate and adaptive immunity. Innate immunity provides rapid protection against infection and is the body's first line of defense. However, it recognizes broad patterns shared by many microbes and does not generate long-term memory. Adaptive immunity is different because it recognizes specific molecular targets. These targets are called antigens, which are molecules capable of triggering an adaptive immune response. Importantly, immune cells usually recognize only a small portion of an antigen, known as an epitope. This specificity allows the adaptive immune system to distinguish among different pathogens with remarkable precision. We also discussed the importance of distinguishing self from non-self. The immune system must recognize harmful invaders while avoiding attacking against the body's own tissues. Finally, we saw that innate immunity uses pattern recognition receptors to detect common microbial features, whereas adaptive immunity relies on highly specific receptors that recognize individual epitopes. The major advantage of the adaptive immunity is that this specificity provides the foundation for immunological memory, allowing future responses to be faster and more effective. In the next lecture, we'll examine the cells responsible for adaptive immunity, B cells and T cells, and see how they generate highly specific immune responses through clonal selection and clonal expansion. With that, thank you so much for attending this lecture, and I'll see you in the next one.